The overall goal of the following experiment is to measure protein secretion from individual cells with high temporal and spatial resolution. This is achieved by lithographically fabricating gold plasmonic nanosensors on glass cover slips for the label-free imaging of single-cell secretions. As a second step, the sensor chip is cleaned and coated with a self-assembled monolayer and then functionalized with ligands that have a high affinity for the secreted analyte proteins. Next, cells are integrated onto the functionalized sensor chips in order to measure their secretions in space and time. The results show that protein secretions from individual cells can be mapped both spatially and temporally without the need for labeling.
This method can help answer key questions in the field of intercellular communication, such as how paracrine and autocrine signaling pathways govern cell motility.
To begin this procedure, deposit a 10 nanometer chromium-thin film on previously cleaned cover slips by electron beam evaporation to avoid charge effects during the patterning and imaging of nanostructures. Following this, spin the first layer of bilayer resist consisting of ethyl lactate methyl methacrylate copolymer at 2000 rpm for 45 seconds. Then bake the substrate at 150 degrees Celsius. After allowing the substrate to cool to room temperature, spin the second layer of polymethyl methacrylate at 3000 rpm for 45 seconds and bake the substrate at 150 degrees Celsius. Pattern the bilayer resist using electron beam lithography, or EBL. Develop in isopropyl alcohol, or IPA, methyl isobutyl ketone, or MIBK.